I'm going to be talking to you about some research that we've done at Stanford with colleagues from elsewhere about studying the community of human microbiome researchers. So they're people, in other words. And so I'd just like to acknowledge some of my colleagues up front. Pamela Sankar at the University of Pennsylvania Center for Bioethics is a co-principal investigator of this study. And the ubiquitous and peripatetic David Relman is a co-PI and been very thoughtful on this project, as well as some of the graduate students that we've had, Angie Boyce at Cornell, Martine Lappé, and Kate Darling at UCSF, and with funding from NHGRI. So the overall goals of our study were to encourage biomedical scientists to incorporate ethical and societal considerations into their research design. But how we were doing that was to kind of take a roundabout route by identifying how scientists talk about and conceptualize risk and benefit as a tool for linking abstract concepts of values and social implications with specific features of research and research design. Because after all, the concepts of risk and benefit are values laden. They're essentially values questions. So our approach was to examine how risk and benefit are portrayed in two different ways. One is to look at both scientific and lay media, and also to assess how microbiome researchers characterize risk and benefit in essentially focus group type interactions. I'm going to talk to you mostly about our work on the scientific articles, which is not quite yet finished, so this is a preliminary assessment of it, as well as a preliminary assessment of some of our focus group data. And we are just starting to do our lay media analysis, which will compare with the presentations of risk and benefit in the scientific articles. So I'll be talking to you today about 270 articles that we selected from the literature that were used, that were selected using terms such as microbiome, metagenomics, microbial genetics, and so forth, as well as two different discussions with microbiome scientists. The first one was a group that was funded by the HMP Demonstration Project, which Lita mentioned, and the second was a group of conducting microbiome research that happened to be in the Bay Area nearby us. So some of the themes from the articles that we pulled out so far, looking specifically for statements about risk and benefit. I'm just going to talk to you about the risk issues today. But one of the things that was striking to us was a near absence of any statements about the risk of microbiome research or its applications, which is not what we were necessarily expecting, but there are so few of these, I can actually show you all four of them, which I will in a minute. What we did see was in the types of risk statements where risk was defined very broadly was what we're calling epistemic risk. And by that we mean risks that people talked about in their scientific articles that were risks to the production of knowledge, so to the conduct of science, so limitations of the tools and methodologies that we have. And I'll just note here that when we've done similar studies in other fields other than microbiome research, we don't see this. So this is unique. And I think it's a sort of illustration of or an indication of the emerging nature of microbiome research as a field. So limitations such as lack of bioinformatics tools to process tons of data, or in one of the articles it was referred to as tsunamis of data. Or the fact that when you're isolating DNA from 16S or whatever, that you're selecting and maybe preferentially selecting. So lots of comments about sort of uncertainty about how to do the analysis and what's the best way. Other risks that people talked about in their articles was risks to humans and others that were addressed by human microbiome research. So not caused by the research or its applications, but addressed by the research. So cancer is a risk, and microbiome research is going to solve that risk or eliminate the risk. Or antibiotic resistance as a risk, which we talked about a lot yesterday. And finally, a tiny number of statements of risks from manipulating the human microbiome. So these are the four statements I was telling you about. So overall, I think related to what we were talking about yesterday, there were a lot of statements that indicated that there were very fluid boundaries between us and the environment. So as we talked about a lot yesterday, bacteria as the environment and also as part of us. In addition, genes as risk in the environment. So this is a little different. It sort of turns the idea of gene-environment interactions on its head when you talk about genes as the environment. I'll give you examples of the quotes in a second. Also, the idea of interaction with toxins through indirect effects outside the body, which was also mentioned yesterday. So these are just illustrations of some of those concepts of how the term environment is used. And someone, I think, mentioned, maybe Steve mentioned yesterday, that everyone has their own idea of what the environment is. And I think one of the things that we've seen in these articles is that there's this fluid definition of environment. And I think that's important in terms of thinking about policy and regulation, because if you can't identify terms like risk or terms like environment, that makes things very difficult to kind of pin down. So I'm just going to read you part of these quotes because they're kind of long, and just focus on the highlighted parts in black here in bold. So one of the articles talked about resistance genes residing in the environmental reservoir do pose a threat to human health. So here you can see they're talking about genes as being the environment, which is not usually the way we think about these things. This quote says, "Could the microflora of the human colon, normally considered innocuous or beneficial, be playing a more sinister role in human health?" So this is, again, the concept we were talking about yesterday, where sort of the bacteria are your friend, but they're sort of your frenemy. In these statements, these talk about sort of the switching back and forth of the microbiome as being sort of beneficial at 1 minute, and then the next minute being virulent or pathogenic. And so it's hard to sort of pin down the microbiome in a single characterization as either being beneficial or risky, because it changes back and forth. So colonizing pathogens one moment and switch to highly virulent pathogens the next. Or most are also commensal species, but they can become pathogenic in responses to change in the environment. This is a long quote, so I won't read you all of this, but this one first talks about the complexity of relationships and the physiologic and genomic flexibility of individual species, demanding a more sophisticated perspective, which I think we'll talk about more later. But it also talks about how we define a relationship, therefore, depends on how we measure benefit and detriment, as well as the breadth and accuracy of our perceptual abilities. So here it's talking about thinking about how we define risk and benefit as being important. And also some of the other quotes and also discussions that we had with microbiome researchers highlighted similar things. I think also briefly touched on yesterday, which was the importance of the issue of scale in thinking about risk and benefit. So how you define risk and benefit depends on whether you're looking at a genomic scale or an evolutionary scale or an ecosystem scale. And everything sort of depends on and is only generalizable to the scale that you choose. This quote talks about resistance genes again, and says that the introduction and progressive accumulation in the environment of antimicrobial agents, detergents, disinfectants, and residues from industrial pollution as heavy metals contributes to the evolution and spread of such resistant organisms in the water environment. So here it's talking about sort of the interaction in the environment as the risk. And finally, this other quote that says, "Certainly it is difficult to believe why public health officers and ecotoxicologists have failed for more than a century to seriously propose the absolute need of preventing the mix between microorganisms from human, animal, and environmental compartments." So a little bit of a soapbox there, but clearly a statement about the environment here and risk from the environment. So I think what you can see here, and what we also heard yesterday, was that the human microbiome has multiple roles. And so it's hard to identify how you sort of assign risk when it has these multiple roles. So I think it might be useful to think about when it's playing these different roles in order to think about when or how to deal with it as a risk, because as we've seen, it serves as both a modulator of risk, but also is a target of risk as well as a source of risk. So it can be all of those things all at the same time. So I'll show you the 4 statements about risk from research or its applications right now, and they were all about probiotics pretty much. So this one talks about risk, specifically an incident of bowel ischemia, and says, although exceedingly rare, these negative effects associated with probiotic administration should raise our vigilance to the possibility of biologically plausible phenotypes that may be harmful. This one says, thus, careful attention needs to be paid to long-term effects of pre- and probiotics, which include the development of infections, cancer, obesity, and immunity. So it's also talking about if the microbiome can affect all of these things, there may be some risk associated with it as well as the potential for treatment. And this one says, this observation raises serious biosafety considerations concerning the use of so-called designer probiotics. And finally, this one mentions sort of food product labels and says, even so, a limited ability to demonstrate clinical efficacy of dietary interventions for disease treatment and prevention has seldom stood in the way of commercial food product viability. So I think we could categorize this in the, as snarky but hard to argue with. Okay, so in terms of communication of risk, I think the scientific publications that we looked at have very little discussion of risk of the microbiome research itself or its applications, or risks discovered in the course of doing microbiome research. And the few statements of risk like that were primarily about the manipulation of the microbiome and risk coming from that. But also, I think there were varied definitions of risk and environment. And just, even though we haven't really finished the analysis of the lay articles, the media, newspaper articles, that were collected or that were published over the same time period as the scientific articles, I can tell you already that the preliminary analysis of those news articles shows that, well, first of all, many of the things published in the lay press are directly reporting on an article that was published in the scientific media. So some of you may have seen the thing about the 3 enterotypes, and that was in the New York Times and other newspapers. So that's typical. But there are also a lot of other articles which are primarily dominated by discussions of probiotics and prebiotics. And there is basically no discussion of risk in those articles about probiotics and prebiotics, at least so far in our analysis. So it doesn't, at some level it does reflect the scientific literature, but at another level it doesn't. So there's a complete, there's a shift in what is discussed or what is selected for publication in the lay media. So I'll just give you a little bit of information from our preliminary focus groups, and there were several themes that came out here. And we had very open-ended discussions in these. And again, I will say that when I've done these kinds of things, asking the same questions to other kinds of researchers, we did not get the same kinds of discussions, completely different discussions. So although these are things that nobody will be surprised by, I will also stress that these are not the same kinds of responses that we got in other talking with groups of scientists from other fields. I think you'll recognize some of these themes from what we talked about yesterday. Some of the issues that came up were funding structures and how that relates to the ability to generate beneficial outcomes from the research. Career considerations, scientific paradigms, and disciplinary perspectives. So especially with regard to the group that we talked about that was funded by a specific RFA for the Human Microbiome Project demonstration projects, there was a lot of discussion about the funding structures, especially because the timeline imposed by this RFA was very short. So you essentially had to have results in about 6 months to do clinical studies that were disease-related. So the researchers said that when you have those kinds of timelines imposed on the research, you have to use opportunistic designs and sampling. So you can't actually choose what you think is the best study design. You have to go with a study that's already in place and use those samples that have already been collected. So they're sort of convenient samples, not necessarily the ones you think are the most appropriate or generalizable. Researchers also told us that funders demand relevance to human health, but that it's also at the same time difficult to get funding for studies that are far along the translational pathway into clinical practice. And another theme that came out was that the grant reviewers demand hypothesis testing, but that animal studies were not allowed in this particular RFA, for example. And then you can't test some things in humans in an experimental way because of ethical reasons, so they felt like they were caught between a rock and a hard place. There were, in other discussions, we had a lot of talk about career considerations. So you can see this quote here, which also specifically mentions risk, but the risk here is being a microbiome researcher. So this quote says, "I think we should acknowledge that microbiome research is a high-risk endeavor from an academic point of view. I'm coming at it from the point of view of an early-stage investigator." Much discussion about this. As we talked about yesterday, there was a lot of mention of complexity versus reductionism. So illustrated by this quote, "We can't sequence our way out of this problem." And a lot of talk about using sort of different approaches, especially ecology and ecological approaches, as well as assumptions about disease causation, which is something we talked about yesterday, the curse of Koch's postulates and so forth, and illustrated by this quote, "What is a pathogen?" And particularly this notion of not being able to distinguish at this point in microbiome research what is a state of health and what is a state of disease, and that there needs to be more research up front that looks at those different concepts in a completely different way than we typically do. So this quote says, "I called my program officer and he asked me to explain it. I explained it to him. He's involved in the Human Microbiome Project, and he kind of said, 'Well, what's your disease?'" And another researcher said, "Folks have had to bring in a disease aim earlier than they might, or maybe at all, when they might not have been interested. So a health-only approach in microbiome research has been the most difficult to get funded and see embraced by the larger community," which to me has been incredibly odd and paradoxical given, you know, like the name's NIH. Okay, and finally, the disciplinary perspectives. There's a lot of talk about this and its effect on being able to think about questions of value, risk, and benefit, and that bringing in different disciplinary perspectives would be useful, not just to conduct the microbiome research, but that other perspectives or other disciplines may have done some thinking about questions of risk and benefit already in a way that would be directly applicable to microbiome research. So this one says, "Conservation biologists you might think of like the clinicians of the plant and animal world, in that they're thinking, okay, I have this tract of land, I want to conserve it or I want to restore it. What's my endpoint?" So here they're talking about sort of the debate that's gone on for a long time in conservation biology about what is good. It's sort of the analogous to what is healthy, what is a state of health. What are we trying to do when we're trying to conserve something? Because that is a value statement. So the scientist goes on to say, "Well, it would be really interesting to see what conservation biologists deem as worthy of prioritizing, and what are their management strategies for these large ecosystems, and what can we learn as human microbiome researchers from all the research they've done in managing their ecosystems." So I think, again, many of these things we talked about yesterday, but were illustrated in what we found, that there are many scientific paradigms that are very central to the conceptualization of risk that are challenged by the human microbiome, such as distinctions between normal, abnormal, health and disease, pathogenic, non-pathogenic, host, parasite, human, environment, and even the concept of risk and benefit. And that peer review and research funding structures are resistant to these paradigm shifts. Also that the complexity of the microbiome may require a broader range of disciplines for several reasons. And that fundamental questions might need to be asked first, such as what is health, what is disease, what does a healthy microbiome look like, before sort of jumping ahead to the applications to specific diseases, or at least happening in parallel to these more applied kinds of studies. So I think that's it, my slides.